Figure 16 shows the effect of light intensity and temperature on the rate of photosynthesis. Describe the effect of light intensity on the rate of photosynthesis. So you can see light intensity this is one of the simplest things to describe. You can see as light intensity increases, the rate of photosynthesis increases and then levels off. Don't overcomplicate it, it's for 2 marks. These graphs come up a lot. So as light intensity increases, so does the rate of photosynthesis. It levels off when something else becomes a limiting factor. So just to understand that point, what we're saying here, if we divide this graph into 2 points, over here, light intensity is a limiting factor because it controls the rate of photosynthesis. If we were, for example, to stop light intensity at this point here, then it would level off here. If we were to stop it here, it would level off here. So this is, over this period, it's the limiting factor. But after a while, increasing light intensity doesn't bring about any change to the rate of photosynthesis. So we can assume that something else is now limiting growth. That could be maybe temperature, or it could be amount of water, or it could be amount of carbon dioxide, but something else. So that's what we got there. Explain the effect of temperature on the rate of photosynthesis. So explain the effect. All right, so firstly, we have to, again, it's one of those explain questions. So what is the effect? The effect is it increases the rate of photosynthesis and then it decreases it if we keep on increasing it. So why? So firstly, photosynthesis is controlled by enzymes. So when you increase the temperature, you increase the rate at which these enzymes can operate. But if it gets too hot, the enzymes denature. That means their active site changes shape. Remember what enzymes look like. So this is an enzyme, and if it's denatured, its active site is all mangled up, and it can no longer fit the substrate. And that's more fitting as a substrate. It can no longer fit the substrate. It no longer has a complementary shape to the substrate, so it can no longer help carry out that chemical reaction. So there's a lot to say there. So where do the marks come from? Firstly, rate increases until optimum temperature for enzyme is reached. So we're saying that, okay, it increases, but then afterwards, any further rise results in a fall in the rate as enzymes denature. Now, you could make this actually a 4 to 5 mark, 6 mark question if you wanted, but this is only 2 marks, so you have to be very concise with what you're trying to say. Rate increases until optimum temperature. That's optimum. That's where it happens fastest. Okay. Some people make the mistake and say at optimum temperature enzymes denature. Absolutely not. At optimum temperature, that's where enzyme catalyzed reaction occurs best, fastest, after which they start to denature. Any further rise results in a fall in rate as enzymes denature. Okay. Be aware, if you have to explain what denaturing is, you do have to talk about the active site changing shape and the substrate no longer fitting. Okay, a student measured the rate of photosynthesis using algal balls in a laboratory. The tube of algal balls was kept at a temperature of 25 degrees Celsius and was moved to different distances from a light source. The result of this investigation is showed that the rate of photosynthesis is... Okay, so let's just get this right. Firstly, algal balls are basically, if you've seen agar jelly, we just basically trap chlorophyll inside agar jelly. A bit like, I don't know if you've ever seen that bubble tea craze where we basically make these fruity scented sort of agar jelly balls. Well, we're doing the same thing, but we're just putting chlorophyll inside. So this could be a plant, but it's not a plant. This is just algal balls instead. Okay, so what we're doing is we're increasing the distance, all right, here, and we'd expect if we increase the distance, the light intensity basically reduces. So remember, as light, due to the inverse square rule, as light spreads out from its source, it becomes fainter and fainter, less intense, and as such, there'll be less light for photosynthesis. So the results of the investigation showed that the rate of photosynthesis is directly proportional to the distance from a light source. So that would mean that as distance increases, the rate of photosynthesis increases. No, that's not true, so that can't be right. Next one, inversely proportional to the light intensity. So that means as light intensity increases, the rate of photosynthesis decreases. Well, this is talking about distance, and this one doesn't factor in distance, so it's irrelevant. And also, it's not true. If light intensity increases, there'll be more light for photosynthesis. Directly proportional to temperature, well, we're saying that this was kept at 25 degrees Celsius. We're not changing temperature, so that's also relevant. So I hope it's the last one, otherwise I've run out of ideas. Inversely proportional to the distance from the light source. So that means as distance increases, the rate of photosynthesis decreases. Notice what I've done here. I think that's kind of important for these ones, because although they're quite easy in some respect, they can all sound a bit similar, and your brain needs to sort them out to make sure you're making the right choice. So just make a little sketch or note of what's going, and let your brain have a bit of time to figure out what makes sense and what doesn't. So obviously this one makes sense, so we go for that one. Explain how substances are moved through a plant by transpiration and translocation. Firstly, transpiration deals with water only, and transpiration involves basically water evaporating from the leaf. That creates a vacuum here, a suction, if you like, and because water molecules are sticky, they sort of stick together. We say they're cohesive. They'll basically draw up water from the xylem as a continuous column of water, which basically, because the water molecules are sticking together, that creates that lowers the water concentration in the roots, and so water moves in from the soil to the roots, and you get this transpiration stream. So that's how substances, or rather water, moves through a plant by transpiration. Translocation is a little bit different. So at the source where certain things are made, like sugars are made, so sugars are made at the source through photosynthesis, and they can be transported through the phloem. They can be transported bidirectionally. That means unlike the xylem or transpiration, where it just transports up the plant, with sugars and such, we can actually transport in a number of directions, and we need to, because we need to provide different cells of the plant with energy or sugars for respiration to supply energy. So these sugars move from source to sink, for example. That's where these sugars are stored. So in the roots, for example, these sugars can be converted to starch where they're stored, and that's what a potato is basically. So it's basically writing as much as you know about transpiration, translocation. So transpiration is the movement of water from the root through the plant, through dead xylem cells, so you can talk about them being dead, driven by evaporation of water, or transpiration from the leaf, from the stomata in the leaves. You can say transpiration. Flow is one direction and occurs by capillary action. What that means is if some water will naturally just enter the root anyway by due to the sort of stickiness of water, the cohesion of water, in accordance with the cohesion tension theory. So you'll just get this water being drawn up. Cohesion tension theory is the idea that the water molecules stick together, but because the vacuum sucking them, they're always under tension, like a column of water that's sticky or elastic that's being stretched and pulled constantly. Cohesion, sticky, tension being stretched. Okay, moving on to translocation. With translocation, you say the movement of sugars from the leaves through the plant as sucrose, so those sugars are moving as sucrose, through living sieve cells. So if you look at the structure of a phloem, I don't know if I've got one here, the phloem looks a bit like this. You've got these, unlike a xylem, which is just basically hollow tubes of dead cells, the phloem has these kind of sieve plates like this. I'm not drawing this very well, it should be like that. So basically you get sugars moving through these sieve plates, like slowly through these sieve plates. So through living sieve cells, these are called sieve cells with sieve plates here, because they look like sieves, of the phloem. The movement is bidirectional, can move up and down to the sinks in the plant where sucrose is needed. So the sinks are basically places where the sucrose is used or for respiration, or it's stored as starch. So basically, you don't need to write all of this down to get 6 marks. Even if you just wrote a little bit of here and a little bit here, you'd end up getting 4 to 5, maybe even 6 marks. So really give these a go. It's unlikely, if you attempted a 6-mark question, as long as you wrote something relevant, you'd likely to get at least 2 marks. So always